Title of his talk is HCV Vaccines and Neutralizing Antibodies, Lessons from Human Infection. Welcome, Justin.
Thanks, Chris. And thanks for the invitation to talk today. Thanks also to Rajan and the other organizers of the conference. It's my pleasure to follow Manson. Always a hard act to follow, but I'm going to talk a little bit on some of the same topics, but from quite a different, I think, perspective. So the first question that I think there's a consensus now on this question, but should a vaccine induce neutralizing antibodies to protect against hepatitis C? I think that most of us think that the answer is yes at this point. That wasn't always the case. But a lot of that consensus, I think, came from data acquired from natural HCV infection. And so I'm going to talk first today about polyclonal neutralizing antibodies and their role in spontaneous clearance of Hep C. Talk a little bit about human monoclonal antibodies. And I think most importantly, talk about the fitness cost to the virus of resistance to these antibodies. And then I'm going to come back at the end a little bit to talk more about that pseudo-particle panel that Manson introduced in his talk that we developed collaboratively across multiple research labs that I think is going to be a really important tool for the field in general. So all of the work that I'm talking about today is made possible by the BBASH cohort, which is a cohort that was established at Hopkins about 20 years ago now by Dave Thomas, and then maintained and enhanced by Andrea Cox since then. This is a group of people who inject drugs who are followed prospectively from before the time they're Hep C infected. And then despite counseling and other support, some of them unfortunately do acquire hepatitis C infection. And they're followed monthly over the course of that acute infection until the time that about 25% of people spontaneously clear hepatitis C without treatment, and 75% go on to develop persistent or chronic infection. And so, of course, this gives us an amazing opportunity to study the immune responses that are present at this critical junction during acute infection, when some people are clearing infection and others are developing a persistent chronic infection. The other thing I'm going to refer to a lot today, and others have talked about this as well, but what is an HCV pseudoparticle? So just for those who don't know, you actually can co-transfect an HCV envelope protein expression plasmid with a defective HIV or MLV backbone with a reporter gene. And what you get is a particle with an HIV core with the reporter gene, but with hepatitis C E1, E2 proteins on the surface. And these particles do enter hepatoma cells in a receptor-mediated way, and you can measure quantitatively neutralization, for example, by antibodies that block that entry. So using this pseudoparticle technology, clear back in 2014, Bill Osborne and Stuart Ray in my group developed one of the first diverse genotype 1 pseudoparticle panels, which is depicted here on the left. And they used that panel to measure neutralizing breadth of plasma in people in the BBASH cohort who were either about to go on to persistent infection or those who are right on the verge of clearing their hepatitis C infection. And these two groups were time matched for duration of infection. I think that's a very important thing to know. And what they saw was that in these time-matched samples, the people with clearance of infection did have greater plasma antibody neutralizing breadth on average than the people who went on to persistent infection. Now, there's some interesting things to notice in this graph. First of all, there's a group down here that did not have detectable heterologous neutralizing antibodies at all, but nevertheless spontaneously cleared their Hep C infection. And I think, given other data on the importance of T cells in clearance, these may well be individuals who cleared primarily through T cell immunity. There's this group up here, which are people who did develop broadly neutralizing antibodies very early in infection and then went on to clear infection. And we pretty commonly now call these elite neutralizers. There's also this interesting group, this troublesome group of people who also early in infection developed broadly neutralizing plasma antibodies and nevertheless did not spontaneously clear their HCV infection. So one of the things that we're really interested in understanding now is what's the difference between these people with high NEUT who clear infection and these people with high NEUT who did not clear infection. This particular issue is even more, I guess, confusing, you could say, because broadly neutralizing monoclonal antibodies have been isolated both from people who cleared HCV infection and also from people with chronic HCV infection. And so what I've shown here is a crystal structure of the E2 ectodomain, and marked on it some of the most commonly targeted antigenic sites on E2 by various different broadly neutralizing monoclonal antibodies. A lot of these were isolated in Manson Law's lab, in Stephen Funk's lab, some by me and Andrew Fliak in collaboration with James Crow at Vanderbilt. The monoclonals marked with asterisks were isolated from people who cleared infection, and the other monoclonals that are shown were all isolated from people with chronic HCV infection. And so one thing I'll point out is we did recently isolate this monoclonal that we called Hep C108 that does appear to target a distinct or unique antigenic site relative to the other BNAbs that had been described. And it was isolated from someone who spontaneously cleared HCV. And I'll just use that antibody to make the point that I think that we've still really only scraped the surface of understanding the different neutralizing epitopes that are targeted by these different antibodies. You know, we tend to lump them into antigenic sites for convenience, but it's important to notice or to note that each of these monoclonal antibodies targets a distinct epitope. So it may be that targeting one particular residue within that antigenic site may potentially make the difference between clearance and persistence. And that's something that we're continuing to study, and I think it is going to be important to understand. The reason I'm optimistic that we're going to be able to make a lot of inroads on that question is this new approach that we have now to isolate E2 specific B cells. Most of the mAbs that were isolated in the past used hybridoma or phage display technology. And Arash Brakui, who spoke yesterday, really showed the first proof that we could use this E2 staining strategy to isolate HCV E2 specific B cells. And we've since developed a similar assay in my lab. And in our particular version of this assay, we actually use a cocktail of antigenically distinct E2 proteins in order to increase the sensitivity of this pull down. And so this has really massively improved the efficiency of both studying E2 specific B cells from infected people and also isolating monoclonal antibodies. Nicole Skinner, when she was a postdoc in my lab, took advantage of this approach to actually study BCR sequences, to do BCR-seq from some of these BBASH subjects. And she actually selected subjects for this study based upon that Osborne study that I was showing you earlier. In that, she took into account both the neutralizing activity of the plasma of the individual and also the outcome of infection of this individual. So rather than having two groups of clearance or persistence, she actually had four groups that she called high NEUT clearance, high NEUT persistence, low NEUT clearance, and low NEUT persistence. And then she compared the BCR repertoires of these different groups. And the really fascinating thing she saw was that there are public B cell clonotypes within these groups. And her definition of a public clonotype were BCRs that shared both V and J usage and had identical CDR3 sequences. And the most sharing, or the largest number of public clonotypes that she picked up were between 2 individuals from the high NUT clearance group. And these were public clonotypes that were not found at all in the high NUT persistence group, for example. So when you look at this, high throughput and really high resolution, you start to see that there really may be differences in the monoclonal antibodies that are generated in people who spontaneously clear Hep C infection compared to those who don't. Even if looking at the plasma, it looks like both plasma samples are relatively broadly neutralizing. As a control, of course, she looked at the E2 non-specific B cells. And interestingly, the clustering here, or the sharing of public clonotypes, looked to be largely driven by the genotype of virus that the person was infected with. And so, we'd speculate that these may be antibodies that target non-structural proteins rather than envelope proteins. So the next question, Arash Barkhoui brought this up yesterday when he spoke. What we're really looking for, I think, is direct evidence that broadly neutralizing antibodies play a role in clearance of infection rather than just correlations like I showed you with the Osborne study. And so what we've done is follow both the viral evolution and the neutralizing response in people who are either clearing HCV infection or developing persistent infection. And we've done this now in quite a few people. I'm just going to show you one person as an example for simplicity. So this is a subject that we call C117 who spontaneously cleared HCV without treatment. Actually was infected for about a year prior to clearance. And what we did was we measured the plasma neutralizing activity over that course of infection. And we also, with the help of George Shaw at UPenn, did single genome amplification of the virus that was present throughout the course of infection in this person. And with the help of Andrew Fliak and James Crow, we isolated also broadly neutralizing monoclonal antibodies from the B cells of this person right after they had cleared infection. So this is just to show briefly that this person did develop broadly neutralizing plasma over the course of their infection. And by day 388, immediately prior to clearance, they did neutralize the majority of HCV-PP variants in this panel of 19 that we were using at the time. This phylogenetic tree shows the evolution of the E1, E2 genes over time in this individual. And these are color-coded by the time of sampling. So you can see that there was sequential evolution of the virus over time in E1, E2. And they're really fast, and you see these tight bottlenecks as well over time, implying that there was strong selection. And the really cool thing is by day 388, immediately prior to infection, there was only one remaining clade of virus, all these viruses that were very closely related to each other, differing only by a few nucleotides. All of these other variants that had preceded were already cleared, and the virus was just hanging on with this one last clade of virus. Then we looked at where the substitutions were showing up. This is the 5' hemigenome of those viruses. Where were the substitutions appearing? And it turned out almost all of the substitutions were showing up and becoming fixed in the E2 protein. Some were in the hypervariable region, as expected, but a lot of other substitutions were showing up in the more conserved regions of E2, including, this is actually the front layer of E2, which is the CD81 binding site that Manson was describing in his talk. So I mentioned that we had isolated a BNAb, a monoclonal from this person that we called Hep C3, and Andrew Fliak in 2018 in Pamela Bjorkman's lab was able to crystallize the FAB of this antibody in complex with an unmutated E2 ectodomain to show us exactly where this monoclonal antibody was binding. And overall, the binding was quite similar to what Manson had seen with his AR3 antibodies, and it was to the front layer of the E2 protein. So what we're showing here are just the critical binding residues of this Hep C3 antibody on the E2 protein. And we're comparing it to the critical binding residues for the CD81 receptor on the same E2 protein. So you can see that there's an amazing amount of overlap in the critical binding residues of the mAb and this CD81 receptor. So this led us to hypothesize that escape from this antibody may have carried fitness cost for the virus. So the first thing that we did was we looked at whether the changes that appeared in the envelope protein over time did confer resistance to autologous antibodies. So here on the top graph, we're showing binding of plasma antibodies to these sequential E1, E2 proteins. And you can see that the later E1, E2 variants are more resistant to the autologous plasma antibodies than the earlier E1, E2 protein. We just introduced, or then we tested the same thing with this Hep C3 monoclonal antibody, and you see the exact same pattern, suggesting that Hep C3-like antibodies were responsible for a lot of the neutralizing activity or binding activity we were seeing in the plasma of this person. So then we looked at fitness of these longitudinal viruses. Just using pseudoparticles, we could see a significant drop in the ability of these E1, E2 proteins to mediate entry over time, about 100-fold drop in pseudoparticle entry. We also cloned these E1, E2 genes into replication-competent virus to make chimeric HCV-CC and measured the replication capacity of these viruses. And again, you can see that the early virus is quite fit and able to replicate in vitro. But by the time you get to that last virus that was present immediately prior to clearance, the virus is completely replication incompetent. We wanted to figure out why that happened. And so we measured the capacity of these longitudinal E2 proteins to bind to either CD81 or SRB1 that was expressed on the surface of CHO cells. And so when we looked at CD81 binding, we see a pattern that's almost identical to what we saw for viral fitness, which is the early E2 bound quite well to CD81, but the later E2 prior to clearance bound very poorly to CD81. And notably, that was not the case with SRB1 binding. And then when we just introduced substitutions in the more conserved regions of E2, ignoring the substitutions that appeared in HVR1, we also saw the same pattern. We saw this loss of CD81 binding as we introduced those substitutions that happened naturally. And again, saw no effect on SRB1 binding. So again, that was just one person. That paper, we described the same thing in two individuals. We've subsequently done this work with a lot more cleared infections. And a lot of these infections shown on this graph were actually people who repeatedly cleared multiple infections. And interestingly, we saw exactly the same result, which is the infections that were cleared when we compared the early envelope protein to the late envelope protein, we saw this drop in CD81 binding. But when we did the same thing with infections that were not being cleared, compared early to late E2, we saw no decline in binding to CD81. So to put this together, I think this is strong direct evidence that neutralizing antibodies are playing a role in natural clearance of HCV infection. And it's not because they're amazingly potent, perfectly broadly neutralizing antibodies. It's just that they're potent enough and broadly neutralizing enough and target conserved epitopes to the point that escape from these antibodies confers a pretty significant fitness cost to the virus. So, this is an illustration of, driving bison off a cliff to kill them rather than shooting each one individually. And I think that that's an analogy of what's happening with BNAbs in HCV infection. So with the time I have available left, I'm going to talk a little bit about this standardized pseudoparticle panel that, again, Manson introduced. I think that this is really, really important for the field for us to start to use a standardized panel so that we can compare neutralization results between different labs and different vaccines. So to develop this panel, the first thing we did was we looked at all worldwide HCV E1E2 sequences, and we picked out E1E2 clones that we had available and mapped them onto this tree, and selected the E1E2 clones that we thought were most representative of the worldwide sequences. And this turned out to be 75 different genotype 1 to 6 E1E2 clones. When we made pseudoparticles, 65 of these clones produced functional pseudoparticles that we could use for neutralization assays. This is the second time you've seen this graph, but we saw when we tested neutralization of a set of 6 different monoclonal antibodies that there was a wide range of neutralization sensitivity of these different pseudoparticles. And one thing I'll point out specifically is that there was no relationship between the genotype of the pseudoparticle and whether it was neutralization sensitive or neutralization resistant. So the non-genotype 1 PPs were scattered across these neutralization sensitivity tiers. Rather than just looking at the mean or the median neutralization sensitivity of these pseudoparticles, we also looked at the pattern of neutralization of each pseudoparticle by all 7 monoclonal antibodies. And we clustered the different E1E2s based upon their pattern of neutralization by these 7 different monoclonal antibodies. And the really cool thing is that all of these 65 E1E2s clustered into 15 different groups of very antigenically similar E1E2 proteins. And some of these groups included E1E2 proteins from multiple different subtypes. So the clustering, the antigenicity of these E1E2 proteins was not dictated by their genotype. Using all of this data, we were able to narrow this panel of 65 pseudoparticles down to 15 representative pseudoparticles. And when we look at IC50s or neutralization breadth measured with either the panel of 65 pseudoparticles or the smaller panel of 15, we get almost exactly the same result. So we think it's not necessary to test every plasma sample or every antibody against 65 pseudoparticles. Instead, I think that we can test against these 15 representative isolates. And importantly, when we tested neutralization by immune sera, these 15 different pseudoparticles, we see the expected ranking of neutralization resistance that we saw with the monoclonal antibodies that we used to select the isolates. And that's with sera from patients infected with genotypes 1 through 6 HCV. You still see the same ranking of neutralization sensitivity. So to summarize, I think BNAbs play an important role in clearance of HCV infection. Clearance appears to be associated with antibodies that can drive resistance substitutions that reduce E2 binding to CD81. E1E2 antigenicity is not dictated by genotype. I think that's a really important point, both for neutralization testing, but also for selection of vaccine antigens. And last but not least, this pseudoparticle panel I've described is freely available. You can get it from AdGene. You can ask me directly, and I'm happy to share it. So I want to thank my team who's done most of this work. I want to specifically point out 2 people. Nicole Skinner did some of the work I showed. She recently left my lab, unfortunately, and has started her own lab in Chris Walker's group. So a rising star in the field. And Andrew Fliak, that you've all heard of before. Believe it or not, he has not been a PI all these years. He's been a postdoc in Pamela Bjorkman's lab, but he's now starting his own lab in the next few months at Cornell. So keep an eye out for both of those folks. And last but not least, thanks to NIAID for funding all of this work through the U19 and various R grants. Thank you.
Great. Thanks for that talk, Justin. That was fantastic. So we have time for one or two questions. We have one in the chat from Dr. Raju. Is there any source or database to get known BNAbs of HCV, is the question.
That's a great question. Not that I am aware of. We've written a few review articles. Stephen Fung has written a few review articles. Manson's written a great review article that talks about some of these BNAbs. So I'd point to those, but I don't think that there is a database.
Okay. Justin, I'll ask a question. It appears that there are a number of people that just don't make a good neutralizing antibody response. Some ultimately clear, some don't. But is there something different about those viruses or other aspects of other components of the immune response, the helper response? I don't know if you've sort of been able to integrate that type of analysis, but why they don't respond.
Yeah, no, that's a great question. So we've looked a lot at this, and I think there are two things that we've seen. First is that the phenotypes of the B cells are different in people who develop good antibody responses and people who don't. Now, why that's the case, we don't know yet. But we just published a paper recently in PLOS Pathogens where we showed that expression of FcRl5 and PD1 on the surface of B cells was associated with a poor E2 specific antibody response. And then the other thing I'll mention is that we've been doing work lately in this reinfection cohort to actually characterize the envelope proteins in people who developed broadly neutralizing antibodies and people who did not. And there do seem to be antigenic differences in the proteins that these people are exposed to that associate with whether they develop BNAbs or not. And that's a paper that's in review right now.
Okay. Thanks, Justin. Seeing no other questions in the Q&A,
Could I ask one question?
Yes, Jens, please go ahead.
It's just that, Justin, great talk. If you think that the role of broadly neutralizing antibodies in clearing HCV is to drive the virus over the cliff, how can we reflect that in our in vitro neutralizations assays?
I would say we just have to make our best guess. And so, what we've done with this new pseudo-particle panel is we've measured neutralizing activity of plasma samples of people who cleared and people who did not. We'll publish those data. And I think that can be a reference that immune sera could be compared to see how closely they simulate what we see in people who are clearing. If there were an animal model where we could look for development of escape mutations, then that would be another possibility to actually sequence the virus over time and look for development of these substitutions in the CD81 binding site of E2.
Okay, I think we have time for one more question from John Law, the University of Alberta. Do you know the quality of the antibody response in people who do not clear the virus?
Yeah, so it's widely variable. There are some people that have what look like really great broadly neutralizing plasma antibody responses who do not clear. We think that in those folks, one, that they may not be targeting exactly the same epitopes as people who clear, and two, that they may be infected with a different virus that's more able to escape and compensate for escape mutations than virus of people who do clear infection. And so those are things that we're studying now, but we don't know the answer yet.
Great. Thanks, Justin. With that, then I'll turn the session over to my co-moderator, Georg Lauer, to introduce